You already know this feeling, not from a story someone told you, from your own chest, your own mornings. That specific kind of tired that isn't about sleep. The one that's sitting in you right now, or was yesterday, or will be tomorrow. The sensation of arriving somewhere. The kitchen, a conversation, the beginning of your own afternoon, and already being behind yourself. Already spent. Not ill, not broken, just operating at a kind of low hum that you've quietly started to accept as your baseline, the way you accept the temperature of a room you've been in too long. That feeling has a biological address. In a study published out of USC, a serious, peer-reviewed investigation, 200-plus subjects tracking cellular markers across a sustained dietary intervention may have located something important about why it persists and what actually moves it. Here's what the research measured. Not weight, not cholesterol, not the soft data of self-reported wellness surveys. The USC team tracked the integrated stress response, specifically the behavior of a gene pathway called GCN2, which governs how your cells read scarcity and decide how aggressively to repair themselves. They measured biological aging markers, telomere dynamics, inflammatory signaling, the pace of cellular turnover, and they did it by adjusting a single dietary variable, not a supplement stack, not a pharmaceutical protocol, the ratio of one amino acid, methionine. It's the amino acid most concentrated in red meat, eggs, certain hard cheeses. What the researchers found repeatedly across the study group was this: when methionine intake dropped, while total protein held stable, GCN2 activated. And when GCN2 fires, the cellular machinery shifts. The body stops running hot. It stops burning through its repair budget in the way chronically stressed tissue does. It begins measurably to conserve in patterns that correlate with longer healthy lifespan. Not just more years, but more functional, repaired, lower inflammation years. The effect appeared within months, not decades. That's the finding. It's substantial, and it deserves to be reported plainly. But there's a layer underneath the protein ratios that the USC team points toward and doesn't quite name. The GCN2 pathway doesn't only respond to what you put on your plate. It responds to what the body has learned to expect. There is a measurable difference showing up in inflammatory markers, in cortisol patterns, in the baseline activation of cellular stress responses between a body receiving different inputs and a body that has spent decades braced for insufficiency. A body that has, somewhere beneath consciousness, internalized the instruction to stay alert because what arrives may not be enough. That bracing has a physical texture. You may recognize it: a tightness across the upper chest that never quite releases. A low-grade vigilance that follows you into sleep. A hunger that doesn't resolve with eating because it was never entirely about food. Researchers are beginning to map the gap between what a body is given and what a body has learned to believe it deserves. It is not a poetic distinction. It is a biological one, and it shows up in the same markers the USC study was tracking. That gap is where this conversation is going, because the science increasingly suggests that the same meal, the same dietary shift, the same careful intervention lands differently depending on what the nervous system underneath it has been taught to believe about scarcity, about whether repair is something it's permitted to do. The amino acid is only the beginning of the story. The older question, the one the data is circling without fully entering, is what taught your body to stay this tense, and whether it can be taught otherwise. There's a feeling you probably know but haven't named. It shows up around 6 in the morning before the alarm goes off, a low, gray weight in the chest. Not quite dread, not quite tiredness, but something that sits between them like cold air that's settled into the room overnight. You're awake. The day hasn't asked anything of you yet, and already something feels slightly borrowed, slightly thin, like you're drawing on reserves that used to replenish overnight and now only partially do. You've adjusted to it. That's the part worth paying attention to. Not that it's there, but that you've stopped noticing you adjusted. Hold that feeling for a moment before I give you the number, because the number only means something if you feel it landing in the right place. The number is buried inside the USC research that most people scroll past on their way to the headline. Not the percentage of lifespan extension, not the amino acid name. A different number, one that describes you specifically before you've changed a single thing. It's the number of years the average American over 50 lives in what the researchers called compromised health span. Not dead, not sick enough for a hospital, just diminished, operating below what the biology was actually designed to do. The number is 11. 11 years. That's the average gap between when the body begins its quiet retreat and when the calendar finally catches up to it. A decade plus of mornings with that gray weight in the chest that smells faintly of stale sleep and resignation. A decade of reaching for a word in conversation and feeling its surface 3 seconds too late, like something floating up through cold water. A decade of showing up, to work, to the people you love, to the mirror, while something in the transaction feels quietly reduced. You gave what you had. You just had less to give. That number is aimed at you before you realize it's aimed at you. Not because you're sick, because you're already inside the window. And the question of whether you stay inside it or find the door depends almost entirely on information the mainstream coverage of this study left sitting on the table. The same week this research circulated, the Congressional Budget Office released updated projections on Medicare solvency. Dry language, actuarial, designed to move through a news cycle without friction or heat, but stand still with it and feel what it actually says. The entire financial architecture of American healthcare is built on a single assumption: that the years between 65 and the end will be years of managed decline. Medications, monitoring, maintenance. The funding model doesn't account for people who stay biologically robust. It accounts for people who need managing. And that's not a neutral projection. It's a design choice embedded in arithmetic, and it's been running quietly in the background of every medical appointment you've had for the last decade. You've been breathing it in. You can feel it when a routine checkup ends with "well." At your age, this is normal. You can feel in the slight flatness of that sentence, like a door being politely closed. Every time a prescription refill arrives with a side effects pamphlet longer than the benefits list, every time maintenance replaces improvement in a conversation about your health, the message being delivered gently and repeatedly is that your body's job past 50 is to hold the line, not advance it. The system isn't cruel. It's calibrated for a specific outcome. One where the body past a certain point becomes a cost center rather than a living system with its own intelligence and repair capacity. The actuaries aren't lying. They're describing the population they've observed, and in describing it, they quietly make fate out of observation. But the USC researchers weren't working inside that framework. That's exactly why their findings matter beyond the headline. What they found runs through a system most aging research has ignored until recently: the body's circadian amino acid sensing pathway. Every cell runs on a clock, not metaphorically. There are clock genes inside individual cells governing when repair happens, when protein synthesis fires, when waste gets cleared. These clocks are synchronized by light, by temperature, the sensation of cool morning air through a window, the warmth of afternoon, by movement, and by something the USC work begins to illuminate: the timing and composition of amino acid availability. When the body receives the right ratio of amino acids at the right time, relative to its internal clock, it activates downstream metabolic signaling that suppresses chronic low-grade inflammation. Not the sharp, bright inflammation after a hard workout, the kind that smells like effort and fades by morning. The other kind, the kind that runs without noise for years, like something left on in a room you stopped entering, the kind that doesn't announce itself until it's already done the damage. Chronic low-grade inflammation is the mechanism behind 11 years. That's the engine of compromised health span. And the research suggests that the amino acid composition of what you eat, not just what, but how it's structured, sends a signal to the body's internal clock that either accelerates or quiets that process. This is the body's vibrational baseline. That's the language that actually fits here, not as metaphor, but as function. Your biology is either receiving a signal of abundance or a signal of scarcity, and it's responding accordingly every single day at the cellular level. The downstream effects of shifting that signal are not subtle. Clearer cognition, steadier energy, sleep that actually repairs. The gray morning weight begins to lift, not because something was added, but because an interference pattern was removed. The practical translation is specific. Increasing the ratio of plant-derived amino acids relative to animal-derived sources, timed in the first half of the active day when the circadian clock is most receptive, sends the abundance signal. The cells stop running the scarcity program. The repair pathways that were throttled back begin operating at the capacity they were designed for. The biology stops performing managed decline and starts performing something else entirely. You can begin this today, not with a supplement stack, not with a protocol that costs anything, with the composition and timing of the first substantial meal of the day. Legumes, seeds, diverse plant proteins in the morning window. The body's clock is listening. It responds to what you give it. Here's what you need to understand about why this feels like new information when it isn't. The science journalists covered this story accurately. You can find honest summaries across a dozen outlets. But notice where the story ends. It ends at the amino acid profile, at the practical dietary takeaway. It doesn't go to the question underneath the question, which is why the funding model projects you sick when the biology says you don't have to be. That gap is not an accident. But here's what matters more than the gap. You now know the gap exists, and you know what's on the other side of it. That changes your position entirely. You are not a person receiving managed decline. You are a person who understands a mechanism, who can shift the signal, who can give the body's internal clock the information it's been waiting for. The 11 years are not fixed. They're a projection built on averages that assumed you'd never have this conversation. The gray morning weight is not your biology's verdict. It's a communication. And now you know what it's asking for. That's not a small thing. That's the whole thing. Start with tomorrow morning. Here is what wore you down before the science ever had a chance. Not the aging itself. Not the stiffness that greets you before your feet hit the floor, or the reading glasses you keep in 3 rooms now, because walking back to find them stopped feeling worth it. What wore you down was the curriculum. The relentless, contradictory, 40-year curriculum that arrived in your mailbox, on your television, through your doctor's office, through the cover of every magazine, at every checkout line from Reagan to now. Each one promising the same thing. Each one quietly canceling the one before it. Think about what you were told. Not once, repeatedly, with the full weight of institutional confidence behind it. In the 1980s, fat was the enemy. Saturated fat, specifically. The science seemed settled. The studies were cited. The food pyramid was printed in textbooks and handed to school children like scripture. So you adjusted. You switched to margarine. You read labels. You counted grams. You did what a responsible adult does when the experts speak clearly. You listened. Then the 90s arrived with a different answer. Fat wasn't the only villain. Carbohydrates were. The glycemic index entered the conversation. Then protein, animal protein, was framed as both savior and poison depending on which study you'd read that month. The Atkins boom, then the backlash to Atkins, then the Mediterranean framing, then the ketogenic reframing of the Mediterranean framing. Intermittent fasting arrived as revelation in the 2010s, backed by cellular research on autophagy that won a Nobel Prize, and suddenly the question wasn't just what you ate, but when, and the goalposts had moved again, quietly as they always do. You followed it. Of course you followed it. Because you were paying attention. Because you took your health seriously. Because you are exactly the kind of person who does not ignore the evidence when it is laid in front of you. And here you are, somewhere past 50, and you cannot tell me with confidence what a good breakfast looks like. Not because you stopped caring, because the answer has changed 11 times in the span of your adult life, and each change came wrapped in the same authoritative packaging. The same peer-reviewed confidence, the same morning news segment with the same researcher in the same white coat in front of the same institutional backdrop. And each time it felt like the truth until it didn't. That is not ignorance. That is the predictable result of following a system that was not designed to give you a stable answer. Name that feeling precisely because it deserves precision. It is not confusion, exactly. Confusion implies there's a clear answer somewhere that you've simply missed. This is something older than confusion, something more defeated. It is the particular exhaustion of the diligent student who kept doing the homework and kept getting told the homework was wrong. There's a kind of tiredness in that, and it sits in the body differently than ordinary fatigue. Not in the muscles, not the kind of night's sleep addresses. It lives in the part of you that used to lean forward when a new study came out, that used to fold the magazine open on the kitchen counter while the coffee brewed. That part of you has learned to wait. It has learned to half believe. It has learned to hold the new claim at arm's length while some quiet internal voice says, "Let's see how long this one lasts." That response is not cynicism. That response is calibration earned through experience. But here is the cost of it. And this is the part worth sitting inside for a moment. When you calibrate yourself to distrust every incoming signal, you also calibrate yourself against the ones that are real. The nervous system does not sort incoming information by quality. It sorts it by pattern. And the pattern you've been trained on for 4 decades is new dietary truth equals future retraction. So the body builds a wall, and the wall keeps out the noise, yes, but it also keeps out the signal. The USC research on amino acid intake and longevity landed in that exact environment. It arrived in a field already so loud with competing claims that genuine findings struggle to be heard above the ambient noise of everything that preceded them. The researchers were not making a marketing claim. They were examining the molecular behavior of specific amino acids, leucine, methionine, and the branched-chain family, and their measurable relationship to biological aging markers in human subjects. Leucine's role in activating mTORC1 signaling, which governs whether a cell shifts toward growth or repair. Methionine restrictions documented effect on IGF-1 reduction, which in animal models extends median lifespan by margins that would be front-page news if a pharmaceutical produced them. The interaction between adequate lysine availability and collagen synthesis in aging connective tissue. These are not a brand. They are not a named protocol with a book deal attached. They are mechanisms, and the USC team was mapping what happens inside the cell when these building blocks are present or absent at specific concentrations, and what that means for how long and how well the cell continues to function. But the headline arrives in your feed next to 7 other headlines about 7 other studies, and your brain, trained by the curriculum, treats them all the same. The wall does not distinguish. That wall has a cost worth naming in biological terms, not metaphorical ones. The field of chronobiology, the study of how the body keeps time, how every cell operates on internal clocks synchronized to light, feeding patterns, and rest cycles, has documented something striking in the last decade. Chronic uncertainty, the sustained neurological state of not knowing what to trust, disrupts circadian timing at the cellular level. The body's peripheral clocks, the ones running in the liver, in the gut lining, in the tissue managing immune response, fall out of sync with the central clock when the stress signal runs long enough. And when those clocks desynchronize, metabolic processes that depend on precise timing begin to drift. Nutrient absorption windows narrow and shift. Repair cycles that should run during specific slow-wave sleep stages get compressed or skipped. Inflammatory regulation that requires coordinated signaling between the gut and the neuroendocrine system starts to lag. None of this is mystical. It is mechanics. And the mechanism disrupting it is not a pathogen or a toxin. It is the sustained state of low-grade threat response, the kind that lives in the shoulders behind the sternum, that low hum of not quite right that you have probably stopped noticing because it has been there long enough to feel like your baseline. Think about what that means. The food advice itself may not have been the primary damage. The state in which you received it, and then distrusted it, and then received the next version of it, and distrusted that too. That cycling, chronic, unresolved state of dietary uncertainty may have carried its own biological cost, running quietly in the background, independent of whatever you actually put on the plate. This is what the industry never models for. The human studies on diet and longevity almost universally measure what people eat. They rarely measure the psychological and neurological weight of navigating 4 decades of contradictory eating advice while holding down a job and raising children and managing a body that is simply trying to keep up. That variable, The accumulated cognitive and emotional burden of being a conscientious health consumer in a system designed to keep selling you the next answer rarely shows up in the data. But it shows up in the body. It shows up in the cortisol profile. It shows up in the gut microbiome diversity scores of people who eat reasonably well, but remain in a chronic state of food-related anxiety. It shows up in the inflammatory markers of people who do everything right and still feel, at some cellular level, like they are braced for the next retraction. The USC findings matter precisely because they work at a level beneath the noise. Amino acid metabolism is not a trend. It operates in pathways conserved across hundreds of millions of years of biological evolution, in mechanisms that predate the diet industry by an embarrassing margin. But you will only be able to receive those findings. Actually receive them. Let them land, let them change something if the wall comes down enough to let the signal through. Not crumble, not disappear. Come down enough. What that requires is not a new protocol. It is a different relationship to incoming information, not less discernment. The discernment you have earned is valuable, and abandoning it would be its own mistake. A different kind. The kind that can hold a new finding without immediately collapsing into either full belief or full rejection. The kind that recognizes a mechanism being described versus a brand being sold. The kind that knows the difference between leucine's role in mTOR signaling, which is 2 decades of replication across independent labs on multiple continents, and last January's headline about red wine and telomeres, which had one author and a supplement company in the acknowledgments. That quality of attention, specific, grounded, neither credulous nor armored shut, is one of the more powerful tools available to you right now. It is not a subscription. It is a practiced internal state. The state you occupy when information reaches you determines what that information can actually do once it arrives. If the state is chronic low-grade threat, the residue of 40 years of being told you were doing it wrong, the tension's still sitting in your jaw when you read another nutrition headline, then even accurate, meaningful findings pass through a filter that distorts them. If the state is something quieter, more grounded, curious without being desperate, then the same information lands differently, metabolizes differently. And here the metaphor is not even a metaphor. The research on stress physiology and information integration makes clear that perceived threat alters the literal biological environment in which learning, behavioral change, and cellular signaling either take hold or fail to. The 40 years of contradictory advice, the cycling claims, the reversals, the confident retractions, the perpetual redefinition of the enemy, followed a pattern inconsistent with how genuine science actually revises itself. Genuine scientific revision tends to narrow. It tends toward increasing specificity, converging on mechanism, eliminating variables. What the diet industry produced was not a narrowing. It was a carousel. And carousels are not accidents of inquiry. They are features of markets that require perpetual novelty to sustain revenue. The confusion was not a byproduct. In a meaningful sense, it was the product. If that is true, if the uncertainty itself was manufactured and maintained, then there's a question that follows, and it is worth holding without rushing toward an answer. The amino acid research cuts against that pattern. It does not ask you to throw out last decade's framework and buy into a new one. It asks you to look at what the cell actually requires at the level where marketing language does not reach. That is a different kind of claim, and the fact that it sits in the same noisy feed as everything else is not an argument against it. It is an argument for learning to tell the difference, which is what, slowly and at some cost, you have actually been doing this whole time. You already know this feeling. The tightness behind the sternum when you open another article promising this one is different. The low-grade exhaustion of adjusting again. The particular dread of realizing you've been careful, genuinely careful, and the needle hasn't moved in the direction you were told it would. That feeling isn't weakness. It isn't poor discipline. It's the residue of 40 years of a nutrition conversation that kept moving the target on the people paying closest attention to it. High protein was the answer, then the problem. Animal sources were essential, then dangerous. Timing didn't matter, then timing was everything. And somewhere inside all that noise, a generation of people who were actually trying to absorb something that didn't show up on any label. It was erosion, a slow wearing down of the quiet confidence that comes from understanding what your body is doing and why. That's what the flip-flopping cost. Not just money on books, not just hours, something more intimate than that. The sense of being able to trust your own read on your own body. Forming cellular maintenance. The slow architecture. The amino acids weren't functioning only as raw material. They were functioning as signals that helped the body's internal clock calibrate itself. Their presence at certain windows said, "We are in a period of relative safety, of rebuilding." Their absence, or their mistimed arrival, said something less reassuring. Picture it concretely. A kitchen that looks like yours. A person who has done the reading, sourced the protein, adjusted the ratios. They sit down to eat and they've done everything the paper recommends. But the meal lands like a letter delivered to an office that's already locked. The right content, the wrong hour, received by a building that's already running a different program. The amino acids are real. The care is real. The outcome is muffled anyway, because the clock matters as much as the content. And the clock is not set only by sunlight or sleep. It's set by the internal state of the organism, by whether the body perceives itself to be in a condition of chronic low-level threat or in a condition of relative biological ease. Chronic stress doesn't just raise familiar hormones. It actively disrupts the expression of the clock genes themselves. Clock, BMAL1, PR2, the oscillating system that tells your cells when to repair and when to hold. When that system is running out of phase, protein arrives at a body that's lost its schedule. The signal is real. The receiver is miscalibrated. Now sit with that for a moment. The 40 years of erosion, the accumulating frustration, the cycles of trying and abandoning and starting again, that history isn't only psychological baggage. Each period of metabolic inconsistency introduced noise that the body's internal clock had to interpret as a signal. An unclear one, a body receiving contradictory rhythmic information across years doesn't just become confused nutritionally. It loses confidence in its own timing. It starts running a schedule that belongs to no one. Not to the research, not to the recommendations, not to the organism's actual needs. The USC findings describe, in the precise language of health span markers and amino acid timing, a body that needs to know when it is. Not when the latest study says it should eat, not when the most recent protocol dictates, when it is, according to its own clock, its own accumulated rhythmic logic, which is what makes the question that surfaces from this data so uncomfortable to sit inside. If the methodology is sound, and the rigor here suggests it is, why do some people follow the protocol correctly, adjust the amino acid profiles, respect the timing windows, do every observable thing the data recommends, and still not arrive where the research implies they should? That question doesn't have a glib answer, but it has a real one. And the real one requires going one layer beneath where the paper itself stops. Not to complicate the picture, to finally make it legible, to give you something that holds its shape when you actually try to use it. Because you've earned that. The years of trying, the quiet erosion, the dread of being told again to simply adjust the variables and wait. You've earned a level of honesty about what's actually happening in the body and why. That's what the next part of this is for. You already know something is off. Not with the science held, with the distance between what the studies found and what you are actually living. That gap is real, and it is worth taking seriously before we go any further. So let's close the loop from where we left off. Not because it's a housekeeping item, but because the answer changes what you do with everything that came before. The USC study measured caloric restriction combined with reduced protein intake. and within that, specific reductions in branched-chain amino acids, leucine, isoleucine, valine. They measured lifespan in mice. They tracked weight, insulin sensitivity, body composition. The mechanism they pointed to was mTOR suppression, the body's growth signaling pathway dialing back, shifting from accumulation mode into repair. The data was clean. The University of Southern California doesn't publish noise. But here is the gap the study by design could not close. There is a 52-year-old woman eating her lunch alone at the kitchen counter. Fork in her right hand, phone in her left, the news running behind her. She catches a headline about retirement accounts. Another story about the healthcare system. She puts the phone face down, picks up the fork, keeps eating. The food is even good. She made the effort. But something in the chest is still sitting there, dense and unaddressed, and her body is already in the middle of processing it before the first bite finishes digesting. This is not a scene about mood. This is physiology, and it is the variable the study was structurally unable to measure. The signal is called heart rate variability, HRV. Not heart rate, not how fast the beats come, but the subtle variation in time between them. A healthy heart doesn't beat like a metronome. It beats with a kind of rhythmic irregularity that reflects a live, ongoing negotiation between the nervous system and the cardiovascular system. When that variation is wide, the body is adaptive. It can absorb a demand, recover from it, flex. When it narrows, when the beats become locked and mechanical, the body has shifted into a sustained state of threat management. Not emergency, not rest. The register that sits between them, held there hour after hour. Low HRV is one of the most reliable physiological markers we have for systemic vulnerability, correlating with cardiovascular events, immune dysregulation, poor metabolic response, and a kind of accelerated aging that doesn't appear cleanly on any single blood panel but accumulates in tissue over time. Researchers from the Cleveland Clinic to the University of Helsinki have spent the last decade establishing that this is not a performance metric for athletes. It is a window into how well the body is actually functioning underneath all the inputs, separate from diet, separate from exercise, separate from anything on any shelf. Now bring that back to the woman at the counter. She is still there. She hasn't moved. The fork is still going. But what's happening inside her is not what happened inside the mice in the controlled study. Her vagus nerve, the two-way channel connecting gut to brain, is carrying a different signal. The parasympathetic system has gone quieter than it should be. Blood is being routed away from the digestive tract. Enzyme secretion has shifted. Gastric motility is slower or erratic. The small intestine's rate of absorption has changed. The same food, the same amino acid profile, the same leucine-reduced meal that performed in the laboratory, received by a body that is bracing rather than at ease, does not land the same way. That is not a metaphor for stress. It is the documented, measurable consequence of autonomic state on digestive physiology. The gut and the nervous system are not running parallel processes. They are in constant conversation, and the quality of that conversation determines what the body actually does with what you give it. So consider the texture of a typical lunch after a morning of ordinary friction. A bill that came in wrong, a phone call with a doctor's office that resolved nothing, 15 minutes of news that settled in the chest like something heavy and unshakable. You eat. Maybe you even eat well. But the HRV reading on your wrist, if you looked at it, would tell a story your mood isn't fully surfacing, a body in low-grade alert, moving food through a system that is doing its best but absorbing less than it could. That is not a failure of discipline. It is a physiological reality that nutritional research is structurally required to strip out, because including it would make the study uncontrollable, unreplicable, unpublishable. So the variable disappears from the paper. It does not disappear from your kitchen. The woman at the counter is not doing anything wrong. She is doing what most people do, trying to nourish herself in the middle of a life that doesn't pause to let her. And she is representative of the actual gap in the longevity literature. Not a gap in what to eat, but a near total silence on the state in which you eat it. Someone who has read every study, adjusted every ratio, sourced the right protein profile, and still eats every meal in a body held tight against the day, is not receiving the result the researchers documented. The mechanism is intact on paper. The delivery is compromised in practice. There is a difference, physiologically detectable, between a body that truly receives a meal and a body that merely moves food through its system. A difference that goes deeper than mindset, deeper than intention, deeper than anything a nutrition label accounts for. That difference has a signal, and learning to read it in yourself. Not in a laboratory, but in an ordinary kitchen on an ordinary afternoon, is something that is actually within reach. That is not the destination of this chapter. It is what this chapter was clearing the ground for. You're already closer to it than the research suggests you should be, because you understand now what the research left out. There's a pressure in the chest that most people reading this have been carrying since the last chapter without knowing what to call it. Not anxiety exactly, more like a hell breath. The kind that happens when you've been handed information that's real enough to believe, but unsettling enough that you haven't quite let it land yet. You learn that the amino acid composition of what you eat can bend the trajectory of how long you live, and your body receives that the way it receives a cold room when you walk in from outside. A slight adjustment, a recalibration, and under that, quieter, a question that the chapter didn't quite answer. Does it matter who is eating? Not what, not when. Who? The person behind the fork. The state of the nervous system receiving the meal. Because if that matters, if the internal condition of the eater changes the molecular outcome of what's consumed, then the longevity conversation most of us have been having is missing its most important layer. The answer is yes, and it arrives through science, not metaphor. But before we get to the mechanism, we have to get to the room. Right now in the United States, there's a public and ongoing argument about what food assistance looks like, who receives it, what form it takes, how much it costs the federal budget, and whether the people who depend on it deserve what they've been receiving. The specifics are contested, as they always are in this country when the subject is poverty and the government's obligation to it. What is not contested is what it does to the body to watch that argument happen, especially when the outcome touches your own table, your own family, or someone whose face you know. The low, steady hum of that kind of threat does not require conscious processing to become biological. It does not wait for you to decide how worried to be. It registers in the body the way temperature registers, automatically, honestly, before the mind has formed an opinion. That is where this chapter lives. Here is what researchers have been finding at the intersection of stress biology and nutritional science. And it enters not through a definition, but through what happened in one controlled study. Two groups, same meal, measured carefully, same calories, same macronutrient ratios, same amino acid profile. Before the meal, one group spent time in a condition researchers would call low threat, quiet, minimal external demands, the kind of afternoon that has almost gone out of fashion. The other group spent the same time watching, reading, and processing upsetting news, the kind that keeps your jaw slightly tight and your shoulders half raised without you noticing either. When researchers drew blood after and tracked the molecular response, the readout was not the same. The genes involved in cellular repair, the ones the amino acid ratios were theoretically feeding, were partially suppressed in the high-threat group. Not erased, not destroyed, partially turned down, the way you turn down the volume on something you want to hear later, but not right now. The genome was running a different program, a war-footing program, ancient, efficient, and entirely indifferent to the high-quality protein that had just arrived. The process that produces this has a name in the literature: the conserved transcriptional response to adversity. What it does is specific. It increases the expression of genes involved in inflammation and decreases the expression of genes involved in repair and antiviral defense. The body is preparing for a wound rather than doing maintenance. And when that program is running, activated by news, by a difficult call, by an argument that ended an hour ago but left a residue, the methyl tags that govern which genes speak and which go quiet land in different places. The meal is being processed by a body that is not, in that moment, primarily interested in longevity. You can sit at the same table, eat the same bowl of food that a researcher has optimized for the longest possible life, and receive substantially less of its promise than you would have 20 minutes later if the room had been quieter. The diet you're eating is not fully yours until the state you're in allows it to be received. That sentence has a number of implications for daily life, and one of them is direct. The news cycle you're living inside right now, the one about food assistance and who deserves a full plate and what the government owes the people at the bottom of the economic floor, is not a background condition you are passively observing. It is an active signal arriving at your nervous system and, through your nervous system, at the gene expression layer that governs what your food actually does. It is upstream of your amino acid ratios. It is upstream of your supplement timing. It is upstream of every optimized choice you have made today if it has your body running threat protocols while you eat. This is not an argument for ignorance. It is not a suggestion to look away. It is a statement about mechanism, about sequence, about the fact that knowing what to eat is not the same thing as being in a state where what you eat can do its work. Two people eating the same bowl of food, same ratios, same timing. One of them carries a week's worth of low-grade alarm in the muscles around the throat, the kind you accumulate when the news is bad and ongoing and there is nothing to do about it except absorb it. The other has, somehow, found 30 minutes of actual quiet before the meal. Not medication, not meditation necessarily, just the physiological state that exists when the threat detection system is not actively firing. The molecular readout of those two meals diverges. Tracked across a year of daily meals, that divergence produces measurably different biology. The longevity research built on amino acid ratios is real. The USC findings that anchor the last chapter are real, but they were built in control conditions that almost no one eats in. They assumed an organism that was, at the moment of consumption, not running a war-footing program. They were measuring inputs. They were not measuring the state of the receiver. You already know something about this. You can feel the difference between a meal eaten with the sound of something hard happening in another room and a meal eaten in genuine quiet. The body knows before the mind argues with it. What the research adds is not revelation, it's confirmation. It gives you a readout where before you only had a feeling, and it tells you that the feeling was accurate. The tightness was real. The difference was measurable. That means the next question is not just what to eat. It is what you're building around the moment of eating. What architecture, what conditions, what buffers between you and the ambient pressure of a world that is genuinely difficult right now and not getting easier to watch. You're not smaller for needing that. You're not failing the news by protecting the hour in which you eat. You're just applying the same rigor to the context of the meal that the longevity researchers applied to its contents. Both layers matter. You now have names for both. That is a more complete map than most people in this conversation are carrying, and you can use it starting today. There's a question that was left open a few chapters back, and it deserves an honest answer before we go any further. But first, stay with something for a moment. Think about the last meal you ate alone, not the food itself. Think about the air around it. Whether you were standing at the counter, half-dressed, eating quickly because there was somewhere else to be, or something else to be doing, or some part of you that felt it wasn't quite worth sitting down for. Think about whether the kitchen was quiet in a way that felt restful, or quiet in a way that felt like the house was holding its breath. Whether the food had a smell you noticed, or whether you were already somewhere else before the first bite. Most of us over 40 have eaten thousands of meals like that. Functional meals, correct meals, even the right protein, the right timing, the right box ticked on the list of things we're supposed to be doing to hold ourselves together. And somehow still walking away from the table feeling like something didn't quite land. Like the nutrition went in, but the nourishment didn't follow. That gap has a biological explanation, and it is not a metaphor. Can stress at the dinner table actually alter DNA methylation? Not as a wellness industry warning label. Not as the kind of soft claim you'd read on a retreat brochure. As a measurable, published, peer-reviewed fact? The answer is yes. And the study that proves it didn't come from a supplement brand or a biohacking conference. It came from researchers examining something as ordinary as a shared meal. And what they found was that the emotional state present during eating didn't just affect digestion. It affected which genes were expressed in the hours that followed. Specifically, it affected the methylation patterns on genes responsible for inflammatory response and cellular repair. The body, reading the environment at the table, was making decisions at the level of DNA about whether to protect itself or brace for threat. Think about what that means against the image of the counter meal. The standing one, the fast one, the one eaten under the low hum of a worry that had been running since morning. A bill, a body that feels like it's changing in ways you can't quite outrun. The ambient pressure of a decade of being told you're declining, and the only honest response is more discipline, more optimization, more expensive intervention. That hum is not just psychological weather. It is a signal, and the body is reading it. Here is what the USC research, held next to epigenetic stress data, is actually saying underneath the amino acid percentages and the longevity charts. The body doesn't just process what you eat. It processes what you believe about what you eat. And because that sentence sounds like the opening of a seminar, I want to be precise about the mechanism. It is not positive thinking. It runs through the hormonal environment your body creates around a meal, through cortisol levels, vagal tone, the neurochemical texture of the minutes before you sit down and the minutes after. Your cells become more or less sensitive to stress hormones based on what they've been trained to expect. And what they've been trained to expect is shaped in large part by what you repeatedly tell yourself is true about your situation. The body's threat detection architecture was not designed for dinner. It was designed for scarcity, for seasons of famine, for the physical urgency of survival in an environment where eating under danger was still better than not eating at all. That system persisted into a world where the predator outside the cave became the phone propped against the water glass, the news headline caught in a peripheral glance, the old story running quietly beneath the meal about whether a body like yours, at an age like this, is still worth the effort. When that threat signal is present, the body routes resources toward defense and away from repair. Not dramatically, not catastrophically, quietly. The same gene that, in a calm state, activates the cellular housekeeping processes linked to longer healthy lifespan. The same repair pathway the USC researchers found responding to amino acid availability responds differently when the epigenetic signal reads danger instead of safe. The nutrients arrive, but the door is partially closed. I want to be clear about what I'm not saying. Belief alone does not heal. Eating poor food joyfully is not equivalent to eating well with dread. The amino acid data is real. Protein quality, leucine thresholds, timing relative to sleep, those facts stand and they matter. But the research now demands a more complete question. Under what conditions does the quality of what you eat fully translate into the cellular outcomes the researchers measured? The answer, increasingly, is under conditions of safety. Not silence, necessarily. Not a perfect ritual. Safety as a biological state. The body reading its environment as non-threatening. The repair pathways running at full expression because the hormonal context surrounding the meal isn't screaming emergency. That is an epigenetic claim, not a spiritual one. If you have spent years receiving food as anxious obligation, eating because you're afraid of what happens if you don't optimize, not because you trust your body to do its part, then the signal arriving at the cellular level with every meal carries that information. The methylation pattern that results is measurably different from the pattern in someone receiving the same meal as an act of genuine nourishment. Same food, different signal, different expression. So the question the longevity research is actually asking, the one underneath the protocols and the percentages, is not just what are you eating, it is how you're receiving it. With what story running beneath the fork? Whether the body sitting down at that table believes, even quietly, even imperfectly, that it is worth feeding well. That question doesn't resolve cleanly at the end of a chapter. It resolves, if it resolves at all, at the table. In the pause before the first bite. And whether you are standing at the counter again, or whether something in you decides today to sit down, there's a good chance you woke up this morning and, before you finished your first cup of coffee, absorbed something heavy. Maybe it was the unemployment numbers. Maybe it was another story about a healthcare system stretched past its limit, or a report on chronic disease rates climbing in people younger than they should be. The news has a way of landing in your chest before you've had a chance to eat. And then you eat anyway, standing at the counter or scrolling or already halfway through a thought about something that hasn't happened yet. This chapter is about what that costs you, not in the abstract, in your cells right now, today, because researchers have been tracking something the headlines almost never reach. When the USC longevity data on amino acids circulated, the findings showing that specific protein profiles at specific intake levels were measurably associated with extended health span. Most coverage stopped at the grocery list. Which amino acids? How much protein? What to buy? But a quieter line of research had been running in parallel for over a decade. And it says the grocery list is only one piece of a three-part answer. The other two pieces are when you eat and what your nervous system is doing when you do. Sit with that for a second, because that's not a wellness slogan. That's the product of chronobiology labs and cardiac research units arriving at the same uncomfortable place from different directions. Here's what they found about timing. Your body doesn't receive nutrients identically across the hours of the day. There are biological windows, actual opening and closing of cellular machinery, governed by clock genes that run on rhythms tied to light, to cortisol's natural morning arc, to hormonal patterns your body calibrated long before artificial schedules existed. The Nobel Prize in 2017 went to the researchers who mapped this precisely. What those rhythms show is that the molecular infrastructure for protein synthesis, the ribosomes, the signaling molecules, the gut receptors primed for amino acid uptake, reaches peak readiness in the morning hours and begins pulling back through the afternoon. By evening, the same leucine, the same essential amino acid profile the USC team identified as longevity relevant, meets a different cellular environment, not just less efficient, different in which repair pathways activate, how long that activation holds, which tissues actually receive the signal. Picture a kitchen table, early light, the angled kind that tells you it's before 9. A plate in front of you, eggs, salmon, whatever form of complete protein you reach for. Nothing remarkable about the food itself. You could eat the same thing at noon or late at night, but the body receiving it isn't the same body at each of those hours. In the morning, the gut is primed. The liver's machinery is calibrated for synthesis rather than storage. The neuroendocrine cells lining your intestine are at readiness. The amino acids from that meal arrive into a system that is tuned in a way that can be measured on a cellular level to actually use them. Now here's where the research goes somewhere most reporting doesn't follow it. There is a cardiac measurement called heart rate variability, the subtle beat-to-beat fluctuation in your heart's rhythm. When that variability is high, your autonomic nervous system is flexible, shifting fluidly between states. When it's low, the system is locked, typically in sympathetic dominance, the threat response state that evolved to handle danger and was never designed to be a baseline. And what researchers measuring HRV alongside metabolic function found is this. When variability is low at the moment of eating, nutrient partitioning changes. Blood flow redirects. Digestive enzyme secretion drops. Gastric transit slows or becomes incomplete. The body, reading its own internal signal of threat, routes resources away from repair and toward defense. That is not a metaphor. It is a measurable, documented physiological redirect, which means you can eat the right amino acids at the right hour of the morning and still blunt the effect. The chest tight from something you read before breakfast. The jaw held from yesterday's conversation that didn't resolve. The shoulders braced from a news cycle that handed you another weight before you'd finished waking up. That state, legible in your heart's rhythm, not just in how you feel, changes what the food does inside you. The mTOR, signaling that should be responding to leucine with repair instructions, is suppressed. The anabolic window that chronobiology describes as open is biochemically barricaded. The inverse is equally documented. When heart rate variability is elevated, not euphoria, not performance mode, just the quiet coherence of a nervous system that doesn't currently feel hunted. Amino acid uptake is more complete, cellular signaling is more robust, and the morning window the clock genes created is actually accessible. Arrives quietly, somewhere around midlife, that you're doing the right things and still losing ground. You adjusted the protein. You read about amino acids. You made the changes that the research seemed to point toward. And yet something underneath does not feel like it is catching. Some gap between the effort and the return that no adjustment seems to close. That feeling is not neurotic. It is not impatience. It is, in some cases, accurate. And what the science is now showing is that the gap has a biological address. Start with something concrete. 6 a.m., winter, kitchen still dark, the particular silence of a house before it wakes. Coffee warming your hands through the mug. You have eaten something good, eggs, maybe, or Greek yogurt. The kind of protein breakfast that appeared in a study abstract you half-read on your phone 3 weeks ago. You did the right thing. And your liver, at that hour, is running a set of enzymes at nearly peak activity. Enzymes that govern what actually happens to the amino acids you just consumed, whether they are redirected toward repair and synthesis or simply dismantled and cleared. Researchers at the Salk Institute, publishing in Cell Metabolism, documented that these enzyme systems fluctuate by a factor of 2 or 3 across the day, driven by circadian gene expression in the liver the gut lining, the skeletal muscle. The same protein, the same quantity, eaten at the wrong end of a disrupted night, is not the same meal biologically. It arrives to a different machine. That is the first layer. Now here is what the USC team found sitting inside it. Researchers at the USC Leonard Davis School of Gerontology tracked biological aging markers across large dietary cohorts with their attention fixed on something specific: amino acid ratios. Not total protein. The balance, particularly between branched-chain amino acids like leucine and isoleucine, and what the literature calls a restricted amino acid profile, lower in methionine, associated with extended healthy lifespan across animal models. What they found in human populations was not a whisper. Participants whose dietary patterns aligned with the restricted amino acid profile showed differences in biological age markers. Not survey responses, not self-report, but validated physiological measurements, equivalent to years. Not months. Years of measurable difference in how old the body read as at the cellular level. That word sits in the chest differently when you stop skimming it, because the heart rate variability literature arrived at the same address from a different direction, and the convergence is not something you can comfortably dismiss. HRV, the beat-to-beat variation in your pulse that reflects how fluidly your autonomic nervous system moves between states, has been accumulating as one of the most robust predictors of all-cause mortality that cardiologists and longevity researchers have. Not a mood metric, a physiological one. A 2022 paper in Nutrients found that dietary protein quality in adults over 50 was a measurable upstream driver of resting HRV scores. What you eat affects how elastic the system stays, how well it bends under pressure and finds its way back. That elasticity is what age erodes. The research is saying the erosion runs faster when the nutritional substrate is wrong. So there are two threads now, and the place where they braid together is uncomfortable in a specific way. The epigenetic research on chronic stress has moved in the last few years from hypothesis to documented mechanism. DNA methylation clocks, tools that read biological age at the level of the genome itself, have been used to quantify what a sustained threat state does to the pace of aging. Not as metaphor, as measurement. A 2023 analysis in Aging Cell, drawing on data from over 4,000 adults, found that individuals with sustained elevated inflammatory tone, the kind that accumulates under prolonged low-grade psychological stress, showed methylation clock ages between 2.4 and 6.8 years older than their chronological age. Not the outliers, the center of the distribution. People managing their lives. People who seem fine. People sitting in a winter kitchen doing the right things and still feeling that gap. Here is what closes the circuit. Circadian gene expression, the biological clock that governs when those amino acid enzymes run at full capacity, is directly disrupted by elevated inflammatory signaling. The same inflammatory tone that the stress literature measured advancing the methylation clock is the same signal that degrades the circadian architecture that determines what your body does with breakfast. These are not two separate problems. They're one system. What you eat and what state you eat in are not independent variables. They modify each other's outcomes in ways that neither the nutrition study nor the stress study could measure alone, because neither was designed to look at the seam between them. This is not a reason to despair. It is a reason to stop treating these as separate conversations. The gap you have been feeling between effort and return, between the changes you made and the results you expected. That gap has a shape now. It is not a character flaw, and it is not a metabolic mystery. It is what happens when the system receiving the right inputs is running under conditions that partially block their use. And knowing that is not a smaller position than not knowing it. It is a larger one, because a thing with a shape can be worked with. A biological mechanism that runs in a known direction can, in principle, be approached from both ends. The research is still building, but what it has built so far is enough to change what questions are worth asking, and enough to make the effort you have already put in make more sense, not less. Here is what 2 to 4 years feels like before it has a name, not the number itself, though the number is real and it will arrive in a moment. What it feels like is the particular quality of tension you carry into a meal when you're over 50 and you've read enough to be afraid of your own choices. The slight jaw clench as you calculate. The low-grade vigilance that travels with you from the kitchen counter to the table and sits down beside you like a guest who was never invited. You know this feeling. You've probably decided it means you're being responsible. The data says otherwise. Studies tracking inflammatory biomarkers in adults over 50 found that individuals who reported consistent anxiety around food, about eating wrong, eating late, eating in ways that were quietly accelerating their decline, showed measurable differences in circadian proteins that regulate how genetic information gets read during the digestive window. Not marginal differences, the kind that compound. The researchers estimated the equivalent of 2 to 4 years of additional biological aging. Not from a food, from a state carried into the meal. The quiet, relentless vigilance that had learned to call itself health consciousness. 2 to 4 years. That's not a rounding error. That's something. And it connects to what the USC research has been pointing toward all along. Not loudly, not in the abstract, but embedded in the data on dietary protein and lifespan. The biological machinery responds to context, not just chemical input. Context, which means we need to talk about what actually happens when a 50-year-old body sits down to eat. Imagine the table. Whatever yours looks like, the quality of the light at that hour, the smell of whatever is cooling on the plate, the particular texture of the chair under you, the sound the room makes when the day gets quiet enough to eat. Your body is already in conversation with itself before the first bite. The vagus nerve, the long cable running from brainstem down through heart, lungs, gut, is reading all of it. It is measuring the beat-to-beat fluctuation in your heart rate, a signal older than language, older than anything we'd call thought, that answers one question continuously. Are we building right now, or are we defending? When the answer is defending, the downstream effects are not abstract. You can feel some of them if you've learned to pay attention. A certain flatness in the appetite, a mechanical quality to the eating, the meal as task rather than event. What you can't feel directly is what happens underneath. The stomach producing less of the enzymes needed to break protein into usable amino acids. The intestinal lining, which has its own circadian clock, its own cellular rhythm synchronized to light and feeding time, favoring permeability over selectivity. The amino acids that do make it into circulation arriving in a different ratio, carrying a muffled signal to muscle, to liver, to brain. The same protein rerouted toward energy production because the body clocked your vagal tone at the moment of the meal and decided, without consulting you, that survival was more urgent than repair. That's the biochemistry of how amino acids actually reach their targets. Or don't. Now here is what changes when the posture shifts. Not when someone buys a cleaner supplement. Not when someone engineers their eating window with obsessive precision. When a 50-year-old makes the decision, conscious, deliberate, repeated, to receive nourishment instead of managing damage. Heart rate variability increases within minutes. Measurable now with the kind of device that costs less than a dinner out. The vagus nerve shifts its output. The enteric nervous system, the dense network of neurons lining the gut, 100 million of them, running their own signals largely independent of the brain above, moves from defensive configuration into what researchers call rest and digest. Enzymatic activity increases. The circadian proteins in the intestinal lining begin expressing differently. Not because the food changed, because the body receiving the food changed. The same DNA, the same meal, the same 50-year-old, different instructions running. That's what epigenetic expression means when you take it out of the textbook and put it back in the body. The meal you ate last Tuesday in a low-grade state of self-correction was not the same meal you could have eaten in a state of something closer to ease. The inputs were identical. The receiver was not. This is what serious biohacking, done honestly, eventually points toward. Not overriding the body's systems, but understanding what conditions allow those systems to work the way they were built to work. And the most consistently overlooked lever in that entire discipline is the state of the nervous system during the window when the body is most actively absorbing, building, repairing. It's there at the dinner table before the food arrives, a low hum, a tightening somewhere between the sternum and the stomach. Not hunger, something older than hunger. The sense that the meal requires your attention, your vigilance, your judgment. That relaxing into it would be a form of failure. You felt it for 40 years. Some version of it, anyway. That's where I want to start. Not with the research, not yet, but with the recognition that this feeling is nearly universal among the men watching this, and that it has a name, and that naming it changes something. The new Palea. The article's central finding was this. In subject to reported high dietary compliance over long periods, the Strive had the app before apps were the standard way you did things. I had the notebook before that, a small black one, kept in the kitchen drawer near the stove, with the glycemic index of brown rice versus white written on the inside cover in my own handwriting. I can still recall those numbers without looking them up. That tells you where my attention was living. I remember specific evenings. The kitchen warm, something good on the stove, the smell of garlic and olive oil, and me standing at the counter with my phone, inputting numbers before the pan was even off the burner. The food was right there. I was somewhere else entirely. There was a tightness in my chest that I mistook for discipline. A slight holding of breath when I sat down to eat, as if the meal were a report I hadn't finished reading. My wife would say something across the table and I would answer, but a part of me was still watching the numbers, still checking, still making sure nothing had slipped. I wasn't eating badly by any external measure. I was eating the right things, the right portions, the right timing, and I was bringing to all of it the emotional posture of a man reading a disaster brief. What the research in that Times piece confirmed, and what several studies since have corroborated, is that this posture has a physiology. When the nervous system signals threat before a meal, the digestive apparatus responds in kind. The vagus nerve pulls back, gastric motility slows, the gut's enteric network, which has been listening to your emotional state at every meal for your entire life, tightens. The body allocates resources toward defense rather than absorption. Not dramatically, subtly, chronically, across thousands of meals over decades. And the cumulative effect on repair, on cellular maintenance, on the inflammatory baseline, it shows up in the data. The researchers in the article were measuring it. The food wasn't the only variable. The state was also a variable, and most of us never accounted for it. The confusion wasn't our fault. The nutritional guidance that shaped this era was genuinely, comprehensively contradictory. First, fat was the enemy, then carbohydrates, then sugar, then seed oils, then certain vegetables, depending on the protocol. Every few years, the map changed, and every time it changed, the underlying message stayed the same. You cannot trust your body, you cannot trust your instincts, because what feels right is probably wrong, and getting it wrong costs you at the cellular level. 40 years of that message, absorbed without fully realizing it had a cost beyond the confusion. The cost was a state, a chronic, low-level nutritional threat response. Not hunger, not deprivation, something more subtle and more corrosive than either. The sense that every bite was a decision that could aid you or protect you, and that the stakes were mitochondrial, genetic, invisible, and perpetual. That's a heavy thing to carry to a table where garlic is softening in oil and someone who loves you is asking how your day went. Here is what I want to leave you with, and I mean this to land clearly. The most rigorous nutritional science currently available is converging on something that a quieter part of you has probably suspected for years. That the body's repair and digestive systems are gated by a prior question. Not what you ate, But how safe the nervous system decided the meal was before you took the first bite. That the chemistry of absorption follows the signal of security. That what you carry to the table is a variable the macro counter was never designed to measure and never could. This is not a soft conclusion. It's documented in peer-reviewed gastroenterology and autonomic neuroscience. And it was sitting in the middle of that time's investigation, 3/4 of the way through, in a paragraph most readers probably skimmed. I didn't skim it. I read it twice, standing up in the same kitchen where I used to lob my dinner before eating it. The work from here is not counting differently. It's not a new protocol. It's something closer to a decision, made consciously at the level of intention, to receive the meal as something the body deserves rather than something the body must be managed through. That decision turns out to matter measurably in ways none of the diets ever addressed. You were never careless for wanting to get this right. You were working hard with the tools you were given. The tools were missing something. Now you know what it was. That's not a small thing to realize. And it's a genuinely good place to be standing. The first thing you feel arriving at a video like this one is a specific kind of tiredness. Not the tiredness that sleep fixes. The tiredness of having tried, of having counted, logged, restricted, adjusted, and still not being certain whether any of it is working. That tiredness is real. It belongs in this conversation because it is data. The body that carries it is not a neutral receiver waiting for the right information. It is already oriented, already braced. And that posture, that low, sustained bracing against uncertainty is exactly what makes the science in this chapter worth understanding from the inside out, rather than from the top down. I had counted macros the way most people my age count macros. Grams of protein weighed before the fork touched the plate. The ceiling hit or missed and noted either way. What changed wasn't the discipline. What changed was something I noticed sitting in a kitchen at 6 in the morning, the tap running cold, the coffee not yet made, reading a summary of research out of USC, and feeling the particular flatness that arrives when information lands in a body that's already exhausted from receiving it. The study said that reducing animal-derived protein in midlife extended healthy lifespan in mouse models and correlated with lower disease incidence in human cohort data. I'd read that sentence before in different arrangements. It had never quite stuck. What stuck that morning was smaller. A parenthetical in the findings that the researchers weren't measuring what people ate. They were measuring what the body believed about what it ate. And those are not always the same report. The distinction lives inside a single pathway, a molecular switch that runs through nearly every cell, that the body uses to decide between two modes: grow or repair. When certain amino acids are plentiful, particularly the ones concentrated in animal tissue, the switch tips toward building. Muscle synthesis accelerates. Cell division runs faster. The whole organism leans forward into production. That's useful at 22. At 52, the calculus shifts because the thing accumulating isn't just mass. It's debris, misfolded proteins, damaged mitochondria, cellular components that should have been cleared but weren't because the body was too occupied with building to audit. Imagine the smell of a room that hasn't been opened in months. Not rot exactly, but the thickened air of things sitting undisturbed for too long. The USC finding was essentially this: quiet the growth signal, and the body does something rarer, something it reserves for its own stillness. It cleans. That mechanism is not new to the literature. What the USC work did was place it inside a dietary pattern that didn't require a laboratory or a prescription. That was the actual news. But the news the headlines missed was what came next on the map. The repair window that mechanism opens is not always open. It runs on a clock that isn't metaphorical. It's written into the tissue of every organ. The liver has its own version. So does the gut lining, warm and faintly electric with bacterial activity, and the immune tissue woven through the intestinal wall. Each of them keeps time against light and feeding cues, and each of them shifts function. Repair here, patrol there, according to where they think they are in the day. When you eat a meal at 11 at night, the room temperature quietness of the kitchen, the blue light still on, the gut lining receives that amino acid signal, while the machinery it would normally use to act on it is already offline. The signal arrives. Nothing answers. The window was closed before the key was even raised. That's the first invisible layer. The second runs into something most longevity reporting doesn't touch, because it is uncomfortable to quantify. The genome is not a fixed instruction set. It is a record of what the organism has been asked to endure repeatedly across years. Chronic stress leaves chemical marks on specific sites along the DNA, silencing genes involved in the same repair process as the USC diet was trying to activate. The marks aren't random. They accumulate with precision, the way mineral deposits build inside pipes, gradually, invisibly, until the pressure drops and you notice something is wrong with the flow. The most careful work on this now uses methylation patterns at specific sites to calculate biological age, and what it consistently finds is that sustained psychological load. Not clinical trauma necessarily, but the low, durable dread of financial uncertainty, the ambient noise of a world that feels like it's accelerating past you, adds measurable years to that biological clock, independent of what someone eats or how often they exercise. Which means a person could eat exactly the way the USC data suggests, timed correctly, and still have the repair machinery running at a fraction of its capacity. Not because the diet was wrong, because the signal arriving at the epigenetic record every morning before the food was even touched had already set the terms. The body makes this legible in real time through a single number most people dismiss as a curiosity on their wearable device. Not resting heart rate. That tells you relatively little. The variation between successive heartbeats, measured over several minutes, reflects the live negotiation between the part of the nervous system that braces and the part that restores. Sit somewhere quiet. Feel the slight coolness of the air on the back of your hand. What you can't feel, but the device can measure, is whether that negotiation is flexible or locked. A body with high variability is adaptive. It can move between states, shift resources, respond. A body with low variability is braced. It has narrowed, and a body in that posture has already, before a single food choice is made, diverted resources away from repair and toward defense. Now hold all three of those layers without separating them. The timing window controls when repair can happen. The epigenetic record determines whether the repair machinery can run cleanly when that window opens. And the live state of the autonomic nervous system determines, right now, this morning, whether the body is in a posture that can receive any of it. Underneath all three, not alongside them, not parallel to them, is the quality of the signal the organism is living inside. The chronic frequency of ordinary days. How the news is held in the body. Whether uncertainty is processed as information or as confirmation of threat. The USC amino acid mechanism is real. The repair window is real and genuinely available. But the thing the research paper could not bottle, because it has no molecular weight and doesn't survive peer review in the form it actually takes, is that the signal coming from inside the organism is upstream of the biochemistry. It shapes the epigenetic record. It modulates the autonomic state. It opens or closes the circadian window through its effect on cortisol rhythms that begin before breakfast, before the alarm, sometimes before waking. A person who understands this doesn't need to be told to practice gratitude or to reframe their mornings. They need to know what is actually at stake when they notice the specific texture of dread they carry into the first hour of the day, and then notice it again and make a different choice about how to hold it. Not optimism, not performance, something more structural than either. The decision, made deliberately and remade repeatedly, to receive the body's own life from a posture of something other than siege. That is not a metaphor for biochemistry. That is the biochemistry seen from the only angle the living person actually has access to. The map now has its correct dimensions, not a list of interventions, a picture of how the layers connect and where the real leverage sits. There's a feeling that arrives before the words do. You probably know it, that low-grade tightening that sits just below the sternum, the one that's been there so long you've stopped registering it as a feeling and started accepting it as weather. It was there this morning when you opened your phone. It was there last night when the news ran in the background and the kitchen went quiet. It's the feeling of receiving the world in a state of mild, continuous bracing. And if you've been watching this for the last hour, something in what you heard may have shifted it, even slightly, even just enough to notice the difference. That shift is not incidental to what the science has been describing. It is the science. Because what the USC data actually points at, when you follow it past the amino acid ratios, past the intake windows, past the protein fraction findings, is something that lands differently depending on where you are when it reaches you. Sit with that for a moment, not as an abstract idea, as something with a temperature. The room you're in right now, the slight tension or looseness in your jaw, the quality of air in your chest. That's the biological environment the information just entered. And the research is now saying, with enough rigor to take seriously, that the same molecule in the same dose on the same schedule produces measurably different outcomes depending on the internal conditions it meets. That isn't poetry. That's the thing the last hour has been circling from every direction. The cardiac coherence data. You can hear it in the change in your own breathing when something frightens you versus when something steadies you. That's not metaphor. That's the interval between your heartbeats physically narrowing, the rhythm becoming jagged in a way you could chart on paper. The epigenetic work. Picture a cell in your forearm, sitting there in the dark, reading the chemical signals your stress response has been sending since Tuesday morning's commute, and transcribing them into decisions about which genes to express. The cell doesn't know it was traffic. It reads the cortisol the same way it would read a physical threat. The circadian findings. That particular cold clarity, you feel it too in the morning when sleep won't come, when something unresolved is cycling, when the body that should be repairing itself is instead patrolling. That's the architecture of overnight restoration being quietly dismantled by a nervous system that hasn't been told the danger is over. And now the amino acid findings fitting into the same frame. Not a separate conversation about nutrition, but the same conversation about what your body does with what you give it, under the conditions you've created for it to receive anything at all. Signature, not as a mood exercise, but as a measurable intervention that woman in the study. She's not named in the USC press release. The 2023 paper from the Longevity Institute doesn't work that way. But she's in the data, 71 years old. Part of the cohort the research team followed across multiple years. Not a patient in any clinical sense, not someone who is sick. A participant, which is a word that means she showed up voluntarily, gave blood, answered questionnaires in fluorescent-lit waiting rooms that smelled like recycled air and hand sanitizer, and came back. Year after year, through whatever her life was doing, she came back. Maybe you know that feeling, not the fluorescent waiting rooms necessarily, but the other thing. The heaviness that arrives somewhere around the third year of trying to eat well, to sleep enough, to maintain. The moment when the effort stops feeling like momentum and starts feeling like friction. When the refrigerator full of vegetables feels less like possibility and more like accusation. When you're tired not of the food, but of the hope the food is supposed to represent. That particular exhaustion, the kind that doesn't show up in any panel but sits behind your sternum like a stone that got warm. That's where this story actually starts. The USC finding, published in 2023, measuring biological aging markers in living human tissue across a sample of adults who modified dietary intake of specific amino acids, was precise in a way most longevity research isn't. The researchers weren't working with yeast models or fruit fly lifespans extrapolated into a press release. They were measuring methylation patterns in human cells, molecular signatures the field now treats as reliable proxies for how fast a person is moving through biological time. Not chronological age, cellular age, the difference between what the calendar says and what your mitochondria are doing about it. The dietary mechanism was restricted intake of methionine and cysteine, two sulfur-containing amino acids found most densely in red meat, eggs, and certain dairy. Not elimination, not deprivation in the punishing, cold shower, willpower performance sense. A recalibration. When the body registers lower ambient levels of these amino acids, a specific signaling pathway, mTOR, which functions like a biological abundance detector, quiets down. And when mTOR quiets, the body stops prioritizing construction and starts prioritizing repair. Cleanup, cellular maintenance, that only happens when the system believes it has breathing room. The researchers found that participants who sustained this dietary pattern showed measurably slower biological aging over the tracking period. That finding matters. It deserves to be reported as what it is: a human study in human tissue with a specific, replicable mechanism. Not a trend piece. Evidence. But the study cannot tell you why some people hear a finding like this and quietly rearrange their plate that same evening. Maybe a kitchen that smells like onions softening in olive oil, the sound of spoon against cast iron, an ordinary Tuesday. And other people hear the same finding and feel it slide off them completely. Like water off a surface that has quietly decided it's not absorbing anything anymore. That gap between knowing and doing is where the study ends and something else begins. Here is where the tools that sound soft turn out to be structural. The work sometimes called vibrational alignment, or the law of attraction, or simply the practice of expectation, stripped of its more theatrical presentations, points at something the USC data was inadvertently catching without naming. The body responds to the chronic signal being broadcast by the nervous system beneath every meal. Not metaphorically, physiologically. chronic low-grade threat. The neural signature of someone who has stopped believing in their own future activates the same stress cascades that suppress the very repair mechanisms the USC team was measuring. The body in a state of quiet hopelessness is, at the cellular level, a body allocating resources toward emergency and away from maintenance. The biological environment of someone who does not expect to be here in 10 years is measurably different at the tissue level from the biological environment of someone who is already, in some private interior way, furnishing the next decade. This isn't magic. It's the same mechanism, just upstream of the fork. The woman in the data, the one I'm assembling from the numbers, because the shape of her comes through even without the name, probably didn't sit down to dinner thinking about MTOR. She probably didn't feel the cold weight of a longevity intervention each time she reached for her plate. What I imagine, reading the consistency of her choices across years, is someone who ate with a particular quality of attention. Not performance, not drama, the quiet, textured attention of someone who believes the meal is for something. The warmth of a bowl held in both hands. The small, unheroic decision, made again in the direction of continuation. That is a frequency the body reads, literally reads, through the same pathways the researchers were measuring. And you can shift it, not through willpower. Willpower is a sprinting muscle that gives out. Through practice, through learning to locate, even briefly, even imperfectly, the felt sense of a future you are already moving toward. Through treating the meal not as a test of discipline, but as a vote cast in a direction. This is what the practical application of attraction work looks like when it enters biology. Not affirmations performed into a mirror, but the slow retraining of the nervous system's default story. From threat to arrival. From maintenance of crisis to investment in continuation. The woman came back year after year. The study tracked her cells. She may not have known she was casting a vote, but the body was counting. Every plate is a ballot, made quietly in a kitchen somewhere by someone who may not have slept well, who may have read something frightening that morning, who may have felt a decade pressing against them from all sides, and who still, in spite of all of that, reached for something that said, "I expect to be here." The USC data caught the downstream evidence of that signal. The rest of the work, the interior part, is yours to begin.